2009 from Huazhong Agricultural University. And now she's an associate professor and master supervisor in the College of Resources and Environment, Huazhong Agricultural University. And your research involves the studies of plant nutrient physiology, biochemistry, and molecular biology. And she has published more than 20 papers in Chinese and international journals. And she is serving on the editorial board, member of the Trends in Soil Science and Plant Nutrition. Oh, that's very, very prestigious. Okay.
Thank you. Good morning. Actually, my work is focused on the plant nutrient and molecular biology. I have received the invitation from the organizer, and I'm interested on this one, so I come here, and I learned so much from this conference. Thank you. My work is something different from other speakers, so it's a short story. It's about the effect of glutamine synthetase genes on rice growth and carbon-nitrogen metabolism. I don't want to show much introduction about the rice and the nitrogen. I think everyone knows the rice. We know in the high plants, the inorganic ammonia should be translated to the organic glutamine by GS and GOGAT. GS means glutamine synthetase, GOGAT is glutamate synthetase. So I think we know the importance of glutamine synthetase. This is the first step for the inorganic nitrogen to the organic nitrogen. In high plants, there are two enzymes for the GS. One is cytosolic GS1, and the second is the chloroplastic GS2. And in rice, there is 4 genes encoding these enzymes, 3 genes encoding the GS1, and only 1 gene encoding GS2. Our work is very simple. Our purpose just want to make these genes overexpressed by the 35S promoter, and we transform these genes to the Zhonghua 11 hydroponic rice in China by the agrobacterial method. So our purpose want, if we got the transgenic plants, whether these transgenic plants can have improved nitrogen use efficiency and low nitrogen tolerance, or may big plant with more biomass and large yield, or something like good quality of rice with more proteins and amino acids. This is our simple purpose. However, we didn't got the simple results. Okay. The first one, I want to introduce the glutamine synthetase 1 gene. This one, glutamine synthetase 1.1 and 1.2 is both from the rice, and the GLNA is a glutamine synthetase gene from the E. coli. First, we got the overexpressed plants, overexpressed transgenic plants here. Then we test the physiological parameters of these transgenic plants, and we can find that both GS activities and the soluble proteins increased significantly in 2 GS1 transgenic plants, and both the normal nutrient and the low nitrogen nutrient solutions. And we also test the total amino acids and the total nitrogen content in the plant leaves, and we got the increased amino acids and nitrogen content significantly in the transgenic plants. Also, we check the free nitrate and ammonia in the leaves, and we found that the ammonia increased significantly in the transgenic plants, and however the nitrate is decreased in the GS1.2 transgenic plants and the GLNA transgenic plants, however increased in the GS1.1 transgenic plants. So here these results indicated that 2J2 may have the different roles in the rice. And at the mature stage, we also check the seed, the yield. However, we didn't get the increased yield and seed amino acids. We found that the yield and seed amino acids decreased significantly in transgenic plants. And until now, we found that at the seedling stage, our transgenic plants have the higher nitrogen metabolic level in the leaves. And at the mature stage, there's lower yield and lower amino acids in the seed. So we want to know what happened from the seedling stage and to the mature stage. Then we test some physiological parameters at the tillering stage and the heading stage. Here, these results showed at the tillering, at both the tillering and the heading stage, we found that the plant have showed the decreased growth phenotypes in the transgenic plants. For example, the root length, plant height, root dry weight, and shoot dry weight. For the spot and the photosynthesis analysis in the transgenic plants and wild-type plants, we found that the changed spot value and photosynthesis parameters in the transgenic plants. Also, from the carbon and nitrogen analysis, we found that lower carbon to nitrogen ratio existed in the transgenic plants here. If that means the unbalanced carbon and nitrogen existed in the transgenic plants, we also check the nitrogen 15 nitrogen content in the transgenic plants, and the lower nitrogen content existed in the transgenic plants. Here is the soluble proteins. We found that also the soluble proteins changed significantly in the transgenic plants at both the tillering stage and the heading stage in the root, stem, leaf, and spiculate. This is the soluble carbohydrates. Also, similar results existed in these transgenic plants. Here, so from the soluble proteins and carbohydrates results, we can find that the unbalanced or the coordinated the carbon nitrogen metabolic status existed in the transgenic plants. And we also check the individual metabolites in the transgenic plants. That means something like sugars, amino acids, and organic acids. We found that they all changed significantly in the transgenic plants, and the different patterns existed in the JS 1.1 and the JS 1.2 transgenic plants. Both different change patterns existed in the between the root and the leaves. Then we take the gene expression level. These genes involved in the nitrogen active nitrogen assimilation and the carbon assimilation, such as NR2, JACE, and PPC Rubisco, these genes. From the results, it's a complex results, but however, we can find that different expression patterns of these genes existed between the JS 1.1 and JS 1.2 transgenic plants, also between the root and the leaf, and among the different nitrogen levels. It's a complex gene expression level. Here we can summary for JS1 transgenic plants showed no obvious changes in growth phenotypes and higher nitrogen metabolic levels at the seedling stage. However, these plants showed a poor growth phenotype and unbalanced carbon nitrogen metabolic status at both the tillering and the heading stage. Then, at the mature stage, these plants should decrease the yield and seed amino acids. So we can conclude these genes expression level altered and caused the unbalanced carbon-nitrogen metabolic status in these transgenic plants. Then we found the decreased yield. Okay, the second part is about GES2. GES2, only one GES2 gene in rice. The GES2 gene is mainly expressed in the green tissues, such as the seedlings and flag leaves. Similarly, we construct the GES2 genes overexpression by 35S promoter, and we have got the GES2 overexpressed plants at the T0 generation, and we check the copy number by southern blot. Also, we can find that the GES2 gene in the transgenic plants have 1 or 2 copies. However, as a T1 generation, we planted these plants hydroponically. Then we found that segregation showed some plants showed green and normal phenotype. However, some plants showed yellow leaves and decreased the dry weight. Then we want to know why. What happened in these the yellow plants. Then we check the GES2 expression level in the yellow plants. We found that GES2 genes expression is decreased significantly when compared to the wild-type plants. However, the green plants have the similar or overexpressed GES2 expression level. Also, the decreased GES activity and ammonia concentration existed in the yellow transgenic leaves. Also, we check the soluble proteins. Decreased soluble proteins existed. And the free amino acids in the yellow leaves, we found that there's something changed in the free amino acids. However, the total free amino acids in the leaves did not change. Then we grouped these amino acids to 5 groups. We found that 3 groups of these amino acids decreased significantly, while the other 2 groups amino acids increased significantly. During the growth of these plants, we found an interesting phenotype. When we grow these transgenic plants, we found that if the plant is small, maybe 1 or 2 leaves at the seedling stage, it's normal and no different with the wild-type plants. When they grow, however, it's come to yellow leaves, this is correlation with the GES2 expression level. That means the small green leaves with the GES2 overexpressed, and the yellow leaves with the GES2 decreased. Another phenotype at the mature stage, at the tillary stage, there's big plants with yellow leaves. Then when we found wild-type plants, it's green leaves. All of them is green leaves. And in our transgenic plants, the young leaves is yellow and the old leaves is green. Also, like the young yellow leaves have GES2 decreased, and the old green leaves have GES2 overexpressed. This is opposite with the wild-type plants. Here we can find that at the seedling stage we have green transgenic plants with GES2 overexpressed, and tillering stage have yellow GES2 co-suppressed plants. Also, we want to find that what happened, what's the difference with the green GES2 overexpressed plants and the yellow GES2 co-suppressed plants. Then we do some experiments. Here, we check the growth phenotype at both seedling stage and tillering stage, we can find that at the seedling stage, the transgenic plants and the wildtype plants did not have so much difference between each other. However, at the tillering stage, there's different significant differences between them. Also, we take the carbon and nitrogen content. We can find that in the transgenic plants, we found decreased carbon-nitrogen ratios. Here, also the total nitrogen content. From total nitrogen content analysis, we can find that increased total nitrogen content in the stem. That means maybe nitrogen is accumulated in the stem and transported to the leaves. The soluble proteins and the soluble carbohydrates is the opposite results from this. We can find that at the tillering stage, the soluble proteins is increased in the transgenic plants, and at the tillering stage, they decreased. However, the soluble carbohydrates is decreased in the transgenic plants at the seedling stage, and then at the tillering stage is increased. So these proteins to carbohydrates imbalance is that means carbon-nitrogen metabolism imbalance. We have also we check the individual metabolic at these plants, and most of them change. Similar. We take the gene expression levels. We also can find that the different gene expression level between the root and the leaf, between the seedling stage and the tiller stage. I want to point that here. In this transgenic plants, we mean that GS2 overexpressed at the seedling stage, then GS2 decreased at the tillering stage. However, we can find that in the root, the GS2 is always overexpressed. Even at the tiller stage, there is reached 500-fold when compared to the wild-type plants. We think that this type of overexpression maybe cause much of the energy in the transgenic plants, then lead to the Okay, this is a summary about the part 2. Here, we think at the tillering stage, the GS2 decreased and make the GSNR GOGAT genes also decrease the expression. So accumulated the amino acids and the proteins degraded. Also, the carbon skeletons accumulated to make to inhibit the rubisco PPC, decrease the expression. And in the stem, the nitrogen accumulated. So we think the nitrogen accumulated in the stem can't be transported to the leaves. This may be some part of the mechanism why the yellow leaves existed in our transgenic plants. Okay, it's the acknowledgement. Okay, thank you. Thank you for your attention.
Any questions?
I think
If there's a disbalance between carbon and nitrogen, would it help if you maybe supply more nitrogen?
We think we change the glutamine synthetase expression level. In the first part, it's overexpressed, and at the second part, it's decreased.
But let's say all these enzymes, all what they are is basically they're catalysts, right? They're catalysts for biochemical reactions. But you need substrate. The substrate, of course, it needs nitrogen source.
Yes, yeah.
So can you maybe if nitrogen is the limiting factor, maybe supply more nitrogen to get more yield?
We have done the results under the low nitrogen condition and high nitrogen condition. Yeah, it's a similar results. So we think the glutamine synthetase gene expression level changes then causes the unbalanced carbon nitrogen metabolic status. Yeah.
So have you thought about So you only look at the enzyme level, right? Or mRNA?
Yeah, yes.
Are you thinking about doing metabolics?
Yeah, we have checked the individual metabolics involved in the carbon metabolism, such as sugars and amino acids and organic acids. However, not other metabolics, just these metabolics. They changed significantly.